Hello, this is the second part of our lecture on functional neuroanatomy. Now, you'll remember that last time we talked about what we see when we look at the brain from the outside. We talked about the basic directions that we use to describe things in the brain. We talked about the functions of the four lobes. We talked about the symmetries and the asymmetries and the crossed nature of the processing of information in the brain. And then I also briefly introduced you to this idea of these ways of visualizing the inside of the brain, which we call the planes of section. And that's where we're going to start off today. The focus of today is really going to be looking at the inside of the brain, sort of finding our way around, understanding the different ways that we can look at that. We're going to talk about identifying gray matter and white matter when we look inside the brain. And then we're also going to talk about major divisions in the central nervous system. We've already talked about the four lobes of the cerebral cortex. In this last part, we're going to talk about another way of dividing up the parts of the brain and understanding their function. So we'll start off going over this. We covered this last time. So when we want to look inside the brain, and particularly when we want to look inside a living brain, we use these three views, these three ways of looking inside that are called the planes of section. There are three main ones that are used: coronal, horizontal, and sagittal. And now remember, coronal is the view that you get of the brain if you were to take a slice through the front, just like this. And in an MRI image, it might look something like this. Horizontal is what you get when you sort of slice the top off like this, and it produces an image that looks like this. And then if you make a slice that's 90 degrees to coronal and 90 degrees to horizontal, you end up with this slice, which is called sagittal. Now, these names might be unfamiliar. There are a couple of little mnemonic devices that you can use. Horizontal is pretty easy, right? Because the slice that we take is pretty much horizontal, flat like the ground. Coronal, another corona is a word for crown. So if you could imagine, the crown of the head here, hopefully that helps you remember how this is oriented. And sagittal, you can just remember as being the one that's 90 degrees relative to the other two. Now, an important thing to remember with these planes of section is that these three sections that you see here are just examples. So anytime that you take a slice that has this orientation, whether it's in the front of the brain, or the middle of the brain, or the back of the brain, that is called a coronal section, no matter where it is. And in a few minutes, actually, we're going to see a little movie that basically consists of a single coronal section that we sweep from the front to the back of the brain. Likewise, anytime you take a slice through the brain that's in this horizontal orientation, whether it's at the top or the middle or at the bottom, that's a horizontal section. And likewise for the sagittal. This right here is a sagittal section that we're seeing here happens to be sort of straight down the middle, cutting the two hemispheres of the brain equally, left and right. That's called a mid-sagittal section, but anytime that you take a slice that has this orientation, it's called a sagittal section. All right, now you may have noticed when we look inside, when we look at these images here, that we see structures that have differing contrasts. We see some stuff that's dark, we see some stuff that's light, we see some stuff that's in between. So what is this stuff that we're looking at? Well, what we're looking at here primarily is the difference between gray matter and white matter. So what is that? What's gray matter and white matter? Well, remember that the brain's made up of neurons. Neurons are just cells of the body, and they have two principal parts. They have their cell bodies, and they have axons. And in the brain, we find that the cell bodies, they tend to cluster together, right? So they tend to all want to stick together with one another. And when they do that, when neuronal cell bodies cluster together, we call those parts of the brain gray matter. Now, as we'll talk about in a little bit, we have lots and lots of gray matter in our cortex. Our cortex is basically one big sheet of gray matter that covers the outside of the brain. We also have, however, clusters of cell bodies that are deep inside the brain that you can only really see through an MRI or another method like that. And when gray matter occurs inside the brain, we call those gray matter nuclei. Now, the other part of neurons, the axons, they also tend to sort of want to cluster together or run together. And collectively, when we're talking about a part of neural tissue that consists mainly of axons, we call that white matter. Now, axons, as we'll learn later, can be these very long, extended processes. And when you have a bunch of axons that are all running in one direction, like we see down here, we call those tracts, white matter tracts, when they occur inside the brain. And we call them nerves when they occur outside the brain. Now, these names, gray matter and white matter, they're important for you to know. They are fundamental descriptive terms for the brain. But there's one trouble with them, which is that when we're visualizing the brain, whether it's in an MRI or through some other method, gray matter doesn't always look gray and white matter doesn't always look white. So you have to be aware of that when you're looking at these images and interpreting them. And to give you an example of that, we're going to look here at two MRI images. So these are, MRI stands for magnetic resonance imaging. If you ever hear somebody going and getting a brain scan, this is very often what we're talking about. This is sort of a picture of the inside of the brain that's taken with a scanner. And what you're seeing here is a coronal section through maybe a little bit of, maybe right about here. I think this is probably a good representation of this coronal section. You see another coronal section here also, but what you'll see is that they look very, very different. And as it turns out, there are two sort of flavors, two different kinds of MRI images that we'll often see. One of them is called T2 weighted, the other one is called T1 weighted. Where T1 and where these names T1 and T2 come from, it's not worth going into in this class. I can talk about it in class if you'd like, but it's not worth going into it at the moment. But you should just know these two kinds, T2 weighted and T1 weighted. And what makes them different is that neural tissue and tissue in general shows up very, very differently between them. So let's look at the differences. So first of all, we can identify the ventricles in both of these MRI sections here. And what you notice is that in the T2 weighted MRI, the ventricles, remember ventricles are these fluid-filled spaces, they are bright white, whereas they're dark black in T1 weighted images. Likewise, we know that on the outside of the brain, that's the cortex, and the cortex is made up of gray matter. And we can see that the gray matter here in this T1 weighted image kind of takes on this sort of intermediate gray value, right? So it's not as deep and dark as the ventricles, and it's not as light as this other stuff, which we'll get to in a moment. It's somewhat in between. And we can see that that's generally the case here in the T2 weighted MRI, that the gray matter, the cortex on the outside, is in this intermediate value. It's not as bright as the ventricles, as the fluid in the ventricles, and it's also not as dark as this other stuff here. Now, what is this other stuff in here? Well, as it turns out, these are axons, and in most of these, in fact, are this is white matter. This is axons coming from these neuronal cell bodies here that live in the cortex. And in a T1 weighted MRI, the axons show up as this light gray color, and they're significantly darker in the T2 weighted image. So hopefully this will sort of give you a way of, when you're looking at an MRI, figuring out whether it's T2 or T1. And then once you've figured out whether it's T2 or T1, you can then begin to look at it and try to understand what you're seeing. Where's the gray matter? Where's the white matter? Now, there's another way of visualizing neural tissue. This way here, the MRI, is really good for looking at the brain in vivo. So this is if we wanted to visualize the brain of a living person or a living animal, MRI is a great way to do it. But there's another way that we can do this, which is looking at the brain in what's called a fixed section or a fixed specimen. This is done. This can only be done ex vivo, right? So this can only be done in a brain that's taken from a cadaver or from, perhaps an animal that's being used for research purposes. And the way generally this is done is that the brain is sort of taken out. It is preserved, and there are different methods of preserving it. And then it's cut into very, very thin slices that are then mounted on microscope slides. And then once you've done that, you can impregnate this slice of neural tissue with a dye or a stain that stains or highlights a certain feature or a certain part of the cell. Now, I'll give you two examples here. On the left, we have a picture, I'm pretty sure this is of a human brain, that has been stained with a cell body stain. And what you can hopefully see is that this cell body stain has, of course, created this sort of dark color all around here in the cortex, which is, of course, where there are many, many neuronal cell bodies. And in fact, if you zoom in on one of these gyri here, you'll see that the cortex, when we stain it with this cell body stain, we can actually see layers here in the cortex. And that's going to be something very important. We're going to talk about that in the next video. These sorts of stains can help us understand the structure of the cortex by revealing its layers. Now, if we look just immediately below the surface of the cortex, what do we see here? Well, we see that there's no stain here whatsoever. And what that should probably tell us is that there aren't any cell bodies here, so therefore there must be axons. And if we go over here to the right, we see that that's the case. So these two slices here were taken at about the same place in the brain, except this one on the left has a cell body stain, but this one on the right here has an axon stain. And hopefully you can see that these two are really showing you, very similar structures, but with just sort of complementary and reversed colors, right? Here, the gray matter is very dark because the stain is attaching itself to the cell bodies. Here, the gray matter is very, very light because the stain, instead of attaching itself to the cell bodies, is staining the axons. And likewise, the white matter over here is very, very light. The white matter over here, the axons are very darkly stained. So we can see the complementary structure of gray and white matter here using a cell body stain or using an axon stain. Now, one other thing I want to point out here is that these different stains can be I think can be useful for visualizing different things. So, for example, if we look here in this slice with the axon stain, you'll notice these voids here, these areas that are completely devoid of stain, or maybe have a paler stain than the white matter areas. And as it turns out, so what is it? If it's not being stained with the axon stain, well, there must be cell bodies. That must mean that this is gray matter. And that's in fact exactly what this is. You can see it a little bit here in this cell body stain, but to my eye, it actually shows up a lot better with this axon stain. And this gray matter here that we see, these gray matter nuclei, they're labeled as subcortical nuclei here in the diagram, are basically a group of nuclei. These are part of nuclei called the basal ganglia, and then we also in here have nuclei called the thalamus and the hypothalamus. So, this is another way of visualizing neural tissue, and in particular it's useful for trying to get a sense of the complementary structure here of the gray matter and the white matter. All right, so what we're going to do now is we're going to watch this nifty little video. I'll put this up on Canvas so you can watch it yourself. So this is a little movie, and it's a movie that consists of a coronal slice that's taken through the brain and then swept from front to back So we're just going to take a moment here. We're going to watch this movie and we're going to pause now and then to sort of see, to look at the different features that we can see as we go through. Now, you can tell from this preview image here, hopefully, that this is going to be a T2 weighted MRI. And how do you know that? Well, you know that the gray matter of the cortex has to be on the outside and the white matter has to be on the inside. And we see that the white matter here is darker than the gray matter. And remember, that's a characteristic of a T2 MRI, where the white matter is relatively dark and the gray matter is relatively light. Now, the one tricky thing about this MRI that we're going to see, and you can even see it here, so these voids here, these empty spaces, are the ventricles. And normally in a T2 weighted MRI, remember, the ventricles are bright white. Here they're black, and the reason is that this MRI was actually taken on a brain that had been removed from a cadaver. And what that means is that there's no fluid inside of the ventricles. And it's the fluid inside of the ventricles that would normally show up as being bright white on a T2 MRI. Okay, so let's begin, and we can see now we're beginning at the very front of the brain, sweeping our way back, and at this point here we're really kind of just going through the front part of the frontal lobe. We can clearly see the white matter with this sort of dark gray shading to it, and the gray matter of the cortex on the outside with this lighter shading. And we can clearly see the two hemispheres of the frontal lobe that are divided by the interhemispheric fissure, which hopefully you remember from our lecture on the last video. Now we're going to continue through, and in just a few moments, there are going to be some features that hopefully you recognize. All right, so right about here, you should begin to see some features that you can recognize. The first one, maybe the one that stands out the most, are these empty voids here, these empty spaces. Now, I told you before that if this MRI were taken of a living human brain, these would be bright white. So these spaces here are, of course, the ventricles. We should also be able to see here that this area, this darker shaded area, is now continuous between the two hemispheres, right? So the interhemispheric fissure no longer goes all the way down, clean through from top to bottom. It's interrupted here by this. So remember, this has got to be white matter because this is a T2 MRI. So what's this big white matter bundle here that is connecting the two hemispheres? You should recognize that as the corpus callosum. Okay, now there's a lot to notice. There's a lot going on here. First off, you've probably noticed that the gray matter here on the outside is no longer sort of continuous and unbroken. We've got these two features here, these two, I don't know what you want to call them, little lobules here of gray and white matter. Now, remember, we're sweeping from front to back. So what could we be seeing here? What could we be seeing the tip of here as we're sweeping from front to back and getting about, a third to halfway through? Hopefully you should recognize these as the very tip of the temporal lobe here. And we're at a part of the brain where the temporal lobe in this slice is completely separate from the rest of the brain here. Now, you should also notice that inside the interior of the brain here, we have the white matter and the corpus callosum that we talked about before. But now we have these sort of paler areas located here and here. And what could those be? Well, we should remember that in a T2 weighted MRI, the white matter is dark and the gray matter is a little bit lighter. And we know that deep inside the brain there are gray matter nuclei. So that must be what these are. This must be gray matter. And in fact, these are part of gray matter nuclei called the basal ganglia. Now, finally, one last little feature here. You'll see that these two gray matter nuclei in each side seem actually to be divided by a dark band. Well, what's that dark band? Well, the dark stuff up here is white matter. Well, this then must be white matter too. And in fact, this is a feature called the internal capsule, and it consists of white matter. It consists of axons that are originating up around here, which is about near where the motor cortex is, the part of the cortex that controls movement. And these neurons here are sending their axons down through the white matter, down through the internal capsule, and eventually many of these axons are going to be sent all the way down into the spinal cord, where they're going to be help controlling our movements. So again, in this slice, we can see a lot going on. We can see temporal lobe, we can see the basal ganglia, we can see the internal capsule here. Another term for this is a tract or a projection, this projection of white matter that eventually is going to go all the way down to the spine. And of course, we can see the corpus callosum, which we've been able to see for a little while now. Okay. Now, a few moments ago, we saw that the temporal lobe here was separated all by itself. Now, in this slice, we can see that it's finally joined up with the rest of the brain. Still, however, we see that there's a big fissure here and over here that is separating this part of the temporal lobe from this part of the brain above it. Now, hopefully, you remember from our first lecture, right? And this is this fissure here. Hopefully, you should remember that this is the lateral fissure, and this is exactly what the lateral fissure describes, is that it runs laterally from front to back, and it separates the temporal lobe from the parietal lobe and from the frontal lobe above it. Another thing that we can see here that we couldn't see just by looking at the outside of the brain is that this lateral fissure is very deep. It goes very, very deep, and it's so deep, in fact, that there's even an extra little bit of cortex that's sort of stashed away here, hidden away, that we wouldn't really be able to see from the outside. This little bit of extra cortex here is called the insula, or the insular cortex. All right, now as we move back and back, we start seeing this structure here. This is called the pons, and it's difficult to see perhaps, but you can see still there are tracts of white matter that are coming down through the pons here, again, eventually bound for the spinal cord. The pons is part of the hindbrain, and we're going to be talking about that in just a few moments. And the last thing we're going to talk about here is you see what looks like some gray matter here, and it kind of looks a little bit like cortex, right? It kind of looks a little bit like this stuff, but the texture is much, much different. The folds here are much tighter, and the white matter has this sort of branching structure here. And we see that it's sort of, separated off here. It's down here at the bottom. Now remember, at this point, we've advanced the slice of our brain, maybe all the way to the back here now. Whoops. And hopefully you should be able to identify this part. I'm just going to advance back to where we were. Yeah, okay, we can just do that from here. Let's do that. You can see we're pretty well far to the back of the brain now. Hopefully you should be able to recognize this as the cerebellum. And you'll see that the cerebellum has its own sort of structure, much in the same way that cerebral cortex does. There's gray matter on the outside, white matter on the inside, and if you look really close, you can see these wiggly little things here. These are little gray matter nuclei on the very inside of the cerebellum. Cerebellum, the name cerebellum means little brain, and you can appreciate, why it was given that name, because its structure here mirrors that of the cerebrum, the rest of the brain, in that it has gray matter on the outside, white matter, and then even gray matter inside of that. All right, we'll just finish this out. We'll see everything getting smaller and smaller. We're seeing the last little bit of the gray matter of the cerebellum here, the last little bit of the gray and the white matter of the cortex. And that completes our tour of the brain in coronal sections. Now, as I said, I'm going to post this video to Canvas so you can take a look at it yourself at another time. All right, we're going to finish up here talking about major divisions of the central nervous system. I already talked about the four lobes. Now we're going to sort of divide the nervous system in a different way. And before we do that, I just want to pause here, and I just want to kind of give you a way of thinking about the functional anatomy that we're going to talk about. So why do we have a brain? What does our brain actually do for us? We could sort of divide up what the brain does into a few really major categories. First of all, it controls and organizes our movements and behaviors. It takes in and it processes sensory information. The brain learns, it forms memories of our past experiences. And in addition to all that, it takes care of our body, the basic functions of our body like breathing and digestion. So when we think about functional anatomy, what we really want to do is try to understand how different parts of the brain are all involved in supporting each of these major functions: our movements, sensations, learning and memory, and the regulation of our body. So, in mammals, we divide the central nervous system into 6 major parts, and the parts are named here. I guess it's 7. It's 6 if we include the spinal cord. So we start with the spinal cord. We have the myencephalon, metencephalon, mesencephalon, diencephalon, and telencephalon. Now I'm gonna apologize up front here that these names may be totally foreign to you, totally unfamiliar to you. Definitely they were unfamiliar to me when I first learned them. But these sort of are just kind of words that you have to memorize and names that you have to memorize. A little mnemonic here that I find helpful. I always remember that the telencephalon is at the top, T for top. The diencephalon is next. And then I always remember that mesencephalon, metencephalon, and myencephalon are in alphabetical order as you go from top down to bottom. And then, of course, the spinal cord is at the very bottom. So when you're trying to memorize these, hopefully that little mnemonic helps you. It's a pain in the butt, but it's just one of these things about how the brain is structured that we have to remember. Now, where the heck do these divisions come from? They come from the fact that all mammals and most vertebrates have the same basic parts. And no matter what kind of mammal we're talking about, and whether it's a fully grown adult or whether it's an embryo, we can sort of find these same parts and match them all up one to one. So you can see that sort of illustrated here, that if we take really any mammalian embryo and we look at the shape of its brain as it's developing, we can find tissue that corresponds to the telencephalon, the diencephalon, and all of the other parts. And we can even do so if we look in not just mammalian embryos, but even vertebrate embryos. We can identify most of these major parts, although, however, in another species, so for example, this might be in a bird or this might be in a reptile, we see that we don't have all of the parts. We don't have both a diencephalon and a telencephalon, but we do have this forebrain structure here that, at some point earlier in our evolutionary history, was present in our forebearers and went on to evolve to become the diencephalon and telencephalon. So these names here, these divisions, we use them and we want to sort of know them because they're a way of understanding the brain structure, not just of our brain, but of really any other brain, especially mammalian brains that we can find. All right, now to wrap up, I'm just going to give you a very high-level outline of some of the basic functions of these areas. So the telencephalon here consists of the neocortex, the cortex, which we talked about before, but it also consists of some structures that are deep in the brain. I mentioned the basal ganglia; they're part of the telencephalon too. And it basically, all of the functions of the four lobes that I talked about are functions of the telencephalon. So that's, our control of our movements, sensory inputs, our conscious awareness, our declarative memory, things like that. Going sort of down in the diencephalon, that consists of parts of the brain that we'll learn about called the thalamus and hypothalamus. The thalamus is important for sort of routing our sensory inputs. The hypothalamus is important for regulating a lot of our basic drives like eating and sleeping and stuff like that, as well as as well as sort of regulating our body hormones. In the mesencephalon here, this is also sometimes called the midbrain. One of its important functions are what are called orienting movements of the head and eyes. So these are the movements that you make. Let's say, for example, you heard a loud sound off to your left and you immediately turn and look at where it's coming from. Those sorts of reflexive orienting movements are controlled by the mesencephalon. In addition to that, their mesencephalon also contains most of the dopamine neurons that exist in the brain. And dopamine is a very, very important neurotransmitter we'll learn about a little bit later for things like movement and reward and stuff like that. In the medencephalon, we have the cerebellum, which, as you remember, is important for controlling balance and coordinating our movements. And we also have the pons. That's an important region where we have lots of inputs coming in and out of the cerebellum, sort of hooking up with the other parts of the brain. Second to last, we have the myencephalon. And down here, we have brain regions that are important for some of our absolute most basic bodily functions, like making sure we're breathing and making sure our heart's beating and things like that. And then at the very bottom, of course, we have our spinal cord. This is sort of where we get lots of sensory inputs first coming into the central nervous system and where we have a lot of our motor commands going out. Now, as you remember when I talked about the four lobes of the cerebral cortex, I described them as being like time zones in the U.S., where really they're made up of a bunch of individual little parts. And so what that means is within each of these regions, there's a lot of diverse functions. It's the same here as well. Now, it's hard to kind of get some sort of general principles because each of these regions does so many things. One thing that you can take away here, perhaps, is that as we go from the sort of bottommost portion to the topmost portion, we can see that the sort of level of involvement or the complexity of the function increases, right? So down here in the myencephalon, we're covering the most basic bodily functions, breathing, heartbeat, and then as we go up, we're looking at the cerebellum, which helps coordinate our movements. Now here we have, basic movements for orienting our eyes. Now up in the diencephalon, we have sensory processing, and then up finally in the telencephalon, we have our higher cortical and cognitive functions. So hopefully that kind of helps you understand in general how these things are organized. All right, so that covers everything I wanted to talk about today. Before we finish, we're just going to do maybe just a little quiz, just to see how much you remember. So first off, you should remember that this plane of section that we're looking at here is a sagittal section. And hopefully you remember that it's a mid-sagittal section, which means that we're going straight down the middle, separating the left and right hemispheres equally. Now, we're looking at two different mid-sagittal sections here. One is a T1 weighted MRI, and the other is a T2 weighted MRI. And hopefully you should be able to remember that the T1 weighted MRI has dark ventricles, the fluid-filled spaces are dark, and the T2 weighted MRI, the fluid-filled spaces are bright white. And hopefully that would help you distinguish these two. You should see in this mid-sagittal MRI this structure here, and you hopefully should know it right away as being our corpus callosum. And if I asked you what this stuff is made of, you should be able to answer right away that it's made out of white matter. Axons is also appropriate. Now, because we know it's made out of axons, well, axons, right, are originating from cell bodies and are going, and they have some target somewhere. Where are they going to and from? Well, these are going to and from neurons that are located in the two hemispheres of the cerebral cortex. Remember, the axons, or the corpus callosum connects the two hemispheres of the cerebral cortex and connects the neurons in them. Okay, this is just a summary of the basic learning objectives for today, and that does it. I look forward to talking to you again when we get to the third part of our functional anatomy lecture. Until then, take care.